just looking through the topics and the program, I think we're going to have a fantastic afternoon. I'm certainly looking forward to it. People will know that it's presentations from PhD students within the centre. There's a selection of long talks and flash talks, and actually, because it's not a physical symposium or we cannot do posters, people will hopefully also know that there are a whole bunch of other talks that we've been running on Fridays and will continue to do during the rest of the month. So welcome again. I should mention that University of Göttingen, the Göttingen Heart Centre, is a partner in our BHF Centre, and if there are any students and or staff from Göttingen who have joined, welcome to you as well. And lastly, I just want to thank the people who organised this, because I've had almost nothing to do with it. That's Alex Evitich, that hopefully you can see on your screen there, Sarah, Sylvia Petroni, James Clark. So thank you to the team for organising it, and I'll just hand over to Alex now to take us through housekeeping, which I assume is not going to involve anything about fire alarms. Thank to you, Alex.
Not this time around. Thank you, Ajay, and welcome everyone for joining this symposium today. So yeah, just a quick reminder to everyone to mute and disable their cameras when they're listening to the talks. If you wish to ask questions, please either raise your hand or post a question in the chat, and the chairs in control of each session will be able to answer those. And due to time constraints, we'll only be able to set questions for the 10-minute oral presentations. And if I could remind the chairs also to keep their speakers to time. And just to remind you that there's a public vote at the end of the symposium, so I hope you all stay on this call right up to the end. And for the first prize for either the oral presentation or 3-minute flash talk, there's a first prize of £100, a runners-up prize of £50, and these will be announced after Dr. Anna Casas, our invited speaker from Maastricht, will give her seminar. So without further ado, can I please ask our chairs, Maria Hasman and Ali Zarban, to join?
So the next speaker is Jessica Chandler. The title of Jessica's talk is In Vivo Structural Dynamics of Myosin Binding Protein C. Jessica, could you please share your screen when you're ready? Myself.
Yeah, sorry, 1 second. Can you see this okay?
Yes, thank you very much.
Okay. So hi, everyone. As Maria said, I'm Jessica Chandler, and I'm a final year student in Malcolm Irving's group at Guy's campus. My PhD is focusing on the in vivo structural dynamics of myosin binding protein C. So we all know that the heart is a highly branched network of cardiac cells, known as cardiomyocytes, that contract to eject blood around the body. The sarcomere is the smallest unit of contraction in the heart, and it's made up of interdigitating myosin-containing thick filaments and actin-containing thin filaments that slide past each other during contraction. Contraction is regulated by coordination of the activation states of these two filaments. So in terms of the thin filament, we've got the classical model of activation, in which binding of calcium to troponin causes a conformational change and a shift in tropomyosin on the surface of actin to expose the myosin binding sites. But also more recently, it's been shown that the thick filament is also regulated and can be the myosin heads can be held in an off state close to the surface of the filament backbone, and this state has low ATPase activity. One of the major players in the coordination or regulation of this contraction states is myosin binding protein C, which is the focus of my project. And as you can see here, it's the blue line, but also up here. So myosin binding protein C is a multi-domain protein of the sarcomere. It's formed of, or the cardiac isoform is formed of 11 immunoglobulin or fibronectin-like 3 domains numbered C0 to C10, and also an M-motif linker between C1 and C2, which has 4 phosphorylatable serine residues. C protein is localized to the sarcomere via a really high affinity interaction between the C terminus, domain C8 to C10, with both the thick filament backbone and titin, whereas the N terminus is thought of as more regulatory. So it's been shown in vitro, so using purified fragments of C protein, generally C0 to C2, that these domains bind both the thick filament myosin heads and also the thin filament, and that these interactions can regulate the activation of these filaments. As I said, this work has all been done in vitro, so we're wanting to look at the interactions of the N-terminus of C protein in vivo. So the aim of my project is to determine the distribution of the N-terminal domains of C protein bound to the thick or thin filaments in situ in cardiomyocytes to better understand the filament binding states of the N-terminus of C protein and the structural dynamics of these interactions in the cardiac cycle. And the approach we're taking for this is by developing an in situ intermolecular FRET-FLIM binding assay between the N-terminus of full-length C protein and thick and thin filament components in adenovirally expressed in isolated living cardiomyocytes. So this project has taken 3-step approaches: design, validation, and application, and I'll take you through design and application today. So quickly, what is FRET-FLIM? FRET is forced resonance energy transfer. It's the non-radiative transfer of energy between a donor and acceptor fluorophore when they're close in space, which is generally less than 10 nanometers. You can see here the excitation of a donor molecule results in the electron becoming excited, and it raises to a higher state. It then undergoes some vibrational relaxation and then returns to ground state, either via a radiative process such as fluorescence or non-radiative processes such as FRET. There's a certain number of conditions that need to be met in order for FRET to be efficient, such as the overlap between the donor and acceptor fluorophores must be sufficient. So you can see here, emission from the donor would directly excite the acceptor molecule. Fluorescence, the fluorophores must be less than 10 nanometers apart, as I said, and also the fluorophore orientations or dipoles must be parallel. Alternatively, FLIM, or fluorescence lifetime imaging microscopy, is the technique we use to measure the FRET. So the fluorescence decay of the donor is measured, and this is used to calculate the fluorescence lifetime of the donor molecule in the absence and presence of an acceptor fluorophore. So you can see here a fluorescence decay curve for the donor molecule in blue, and at a certain intensity 1/E, this equates to the lifetime of the donor. In the presence of an acceptor in red, you can see that the fluorescence decay becomes steeper, and this shortens the fluorescence lifetime of the donor in the presence of the acceptor. FRET efficiency can then be calculated by comparing these two lifetimes using this equation. So moving on to my assay that we've developed, as I said, we're adenovirally expressing different sarcomeric proteins in cardiomyocytes. So for my donor, I'm looking at the N-terminus of C protein. So I've got a TFP attached to the N-terminus of full-length myosin binding protein C. I've then evaluated multiple different acceptor molecules on the different filaments. So for the thick filament, I've got an adenovirus expressing M venous RLC. This is a component of the myosin head. And for the thin filament, I have M venous TNT, and this is troponin T, which is a member of the troponin complex. I've also used a number of different actin binding proteins, LifeAct M venous, and also phalloidin ifluor 514. This is just a dye that's very similar to the M venous fluorophore, and these will hopefully stain the length of the thin filament. When looking then in the context of FRET, so we want the fluorophores to be less than 10 nanometers away from each other. Depending on where the N-terminus of myosin binding protein C is, we should hopefully get a FRETable interaction. And then just quickly looking at those fluorophore pairs, we've got the MTFP venous pair and the MTFP ifluor 514 pair, and there's a nice spectral overlap between the two. So moving on to the application of my FRET assay, I'll just quickly take you through the optimized workflow that I've developed. We isolate cells from neonatal rats at day 0 and then infect them with adenovirus and culture them for 3 days. I then skin them with Triton, which is a detergent which will permeabilize the membrane so that we control the intracellular environment of the cells, and wash them into either a relaxing buffer in which both the thick and thin filaments are switched off, or a rigor buffer in which the myosin heads are forced to attach to the thin filament and can't detach. So this is like a forced on state. At this time, I also wash in the Phalloidin iFluor 514 if that's the acceptor I'm using. I then fix the cells and image them by either epifluorescence imaging, where looking at the donor and acceptor intensities, I can determine the expression of the different adenoviruses, and also then perform multiphotron FLIM. I then process the FLIM images, and you can see here the little yellow box refers to the pixel that I've got. And for that pixel, you can see a decay curve, which I fit with a mono-exponential decay. From this decay, this calculates the fluorescence lifetime, or tau, in that single pixel. This can then be done across all the pixels in the image to create a fluorescence lifetime map for the image, where the low lifetime is shown in blue and a high lifetime, which would be indicative of FRET, is shown in red. This can then be converted into a fluorescence lifetime histogram, which I then generate intensity weighted average fluorescent lifetimes for both the donor in the absence of the acceptor and the donor in the presence of the acceptor. I would then convert these into FRET efficiencies and plot the data. So this is the kind of data you'll see. I've now performed a fluorescence lifetime imaging screen for all of my donor and acceptor FRET probe combinations in both skinned NRCs in both relaxing and rigor conditions. So just looking at the imaging, for my donor TFP-C protein, you can see both in relaxed and rigor conditions, the donor intensity, the acceptor intensity, and the fluorescence lifetime image. I've then done this in the presence of different acceptors. So we've got iFluor 514, LifeAct Venus, Venus RLC, and Venus TNT, and also a positive control which has TFP and Venus stuck together by a few amino acids. And you can see even in the fluorescence lifetime images, there's a variation in the colour and some towards the yellow and red spectrum, which suggests a FRET signal. When I've quantified this, you can see my positive control gives me a really nice FRET signal of about 20%. Unfortunately, the TFP-C protein and LifeAct Venus, Venus RLC or Venus TNT combinations didn't give me any FRET signal. This doesn't mean there's no interaction between the two proteins. It just suggests that maybe either my expression levels are wrong or orientation issues are present. However, in more promising news, I did get a detectable FRET signal between TFP-C protein and Phalloidin iFluor 514, which suggests that in situ, at least a proportion of the N-terminus of C protein is bound towards the thin filament. I then wanted to evaluate this a little bit further, and we also did a combination of TFP-TNT with Phalloidin and also TFP-RLC with Phalloidin. So this TFP and Phalloidin is a thin-to-thin filament component, so this high FRET efficiency that we see is really promising. However, for RLC and Phalloidin, we got a FRET signal of about 10%, so this is a thick filament to thin filament interaction, or distance measurement at least. And the fact that for the N-terminus of C protein, I got a FRET efficiency somewhat between the two, suggests that the N-terminus of C protein isn't exclusively bound to the thin filament or to the thick filament, that a proportion is either bound to both at both in relaxing and rigor conditions. So in summary, I have developed and optimized a FRET-FLIM workflow to study the interactions of sarcomeric proteins and the thick and thin filament components in skinned NRCs. Application of the FRET film assay has detected a FRET signal between TFP-C protein and actin filaments stained with phalloidin ifluor 514 in skinned NRCs in both relaxing and rigor conditions, which is intermediate of that detected for TFP-RLC or TNT with phalloidin. And this suggests that a proportion of the N-terminal domains of C protein may associate with the thin filament in situ, and also that the N-terminus of C protein exists as a heterogeneous population, not exclusively bound to the thick or thin filaments in relaxing and rigor conditions. So my next steps are to study these interactions following beta-adrenergic stimulation. And yeah, that's all. I'd like to thank Malcolm and Thomas, my supervisors, as well as everyone else on this list, and yeah, open the floor to questions.
Thank you, Jessica. Very nice talk. So I think we have one question. Alex.
Hi, Jessica. That was great. Really nice talk. I wanted to ask you about the possibility of doing live FRET FLIM. And I'm assuming that, you know, part of the problem with FRET FLIM of the technique that you're using is the photon counting and how long that takes. But I think that Simon Amir Beg within the Randall has developed a live FRET FLIM microscope. So I'm wondering, A, is that possible to use? And B, if you were to go through this permeabilization process and use the phalloidin ifluor 514, is that in any way toxic to living cells, or are you able to see them contract?
So in terms of using the live FRET FLIM, yeah, obviously at the moment my images do take quite a long time to acquire. So I think each image is 300 seconds, so 5 or 6 minutes. So it does take a very long time to get that signal count. But that is just the instrument that I'm using. If I did put it on Simon's more high spec instrument, then maybe we'd be able to push to using the live cells rather than fixed cells. But yeah, also at the moment, in terms of having to use the phalloidin as the alternative rather than having live cells or intact cells, we've had to move to a skinned cells approach. And that you do see some, because I'm putting them in relaxed or rigor conditions, they then sit in kind of as a steady state rather than looking like they're contracting. I haven't really tested whether or not I can put them in a media that contracts them once they've been skinned, but I think, I mean, if the media is around, then maybe. But the phalloidin doesn't seem to be toxic at all. Okay,
thanks.
Any other questions? So I'm just wondering, Jessica, about the localization of your fluorescent proteins in the cells. Can you tell us more?
Yeah. So in the images, let's go back. So you can't really see. Well, let's just go forward. I do have some images. So I did, as I said, I did some validation experiments originally to check whether or not, firstly in vitro, to check whether or not the addition of a fluorophore to the N-terminus of C protein would alter the interactions that had been seen previously, and confirmed that the differences weren't significant. However, I also did some vivo validation looking at the localization of these proteins. And I did this by both confocal and STED imaging. And obviously the STED you can see much clearer. You can see the nice doublets of, this is venous C protein or venous RLC and venous TNT. You can see the nice doublets of C protein. And when I quantified this as half maximal widths of the doublets, this almost kind of matched that of seen in EM, which suggests that my proteins are all localizing correctly to the sarcomere, even though they're being adenovirally expressed or overexpressed. So that shouldn't be an issue.
Thank you very much, Jessica.
I'm now going to share my screen where I'll be showing you a series of 3-minute flash talks. In the interest of time, as I said at the beginning, we won't be able to ask questions. But please do view them all because we want your vote at the end. Thank you.
Hi, my name is Hannah and I'm a second year PhD student in Dr. Alison Brewer's lab. I'm interested in whether TET2 participates in endothelial inflammatory responses. So we know that from one genome we get an incredible diversity of different cell types performing different functions despite containing an identical DNA sequence. This is attributed largely to epigenetics, which are heritable changes in gene expression which don't involve an alteration of the sequence itself. This includes histone modifications and DNA methylation. What these modifications really do is they change the way that DNA is packaged and compacted within the nucleus, making some regions of DNA more or less accessible to transcription factors. In context of cellular stress or any stimulus, the cells can change these epigenetic modifications for dynamic transcriptional reprogramming, which is heritable by daughter cells. I'm interested in the TET family of proteins which demethylate 5-methylcytosine to ultimately replace it with unmethylated cytosine, and this is associated with gene activation. DNA methylation patterns have been observed to be different in cardiovascular diseases and diabetes compared to in healthy tissues, and this may be causal in the disease. An early stage of these diseases is the dysfunction of endothelial cells, which form the inner lining of blood vessels, and part of this dysfunction is to the inflammatory response that they can elicit. In recent publications in other cell types, it's been suggested that TET2 may be involved in resolving or repressing inflammation, and we wonder whether TET2 may also be involved in inflammatory responses of endothelial cells. So an early experiment in my PhD was RNA sequencing of human umbilical vein endothelial cells upon knockdown of TET2. And in these cells, we see that compared to controls, they have much higher levels of expression of interferon-sensitive genes in the pathways responding to type 1 and type 2 interferon. We further looked at this upon the addition of interferon alpha and then looking at time points after its removal to see whether these cells have, which lack TET2 have any impact on the resolution of that inflammatory response. And indeed, we see that in TET2 knockdown cells, there's a higher expression of interferon-sensitive genes, using here an example of IFI-TM1, both upon activation with interferon alpha and following its removal. So this suggests that TET2 may be involved in the suppression of interferon responses in endothelial cells. And now I'm looking at the functional impact this has on endothelial cells and the mechanism by which this occurs, whether it involves TET2 alone or in combination with other binding partners. This work is important in understanding more about endothelial dysfunction, which may lead to solutions to prevent or treat cardiovascular diseases at an early stage. Thank you to the rest of the Brewer Lab, to the British Heart Foundation for funding, and thank you for listening.
Good afternoon, I'm Brenton Barrett from Professor Susan Brain's lab, and today I'll be giving a talk on the vasoconstriction response to cold, which is impaired in diabetic mice. So, why diabetes? Hyperglycemia and diabetes can induce multiple downstream targets that ultimately damages the peripheral neurons. This can lead to the development of diabetic peripheral neuropathy, which can result in an abnormal ability to discriminate temperature. Transient receptor potential proteins, TRPA1 and TRPM8, are cold-sensitive channels which we know to be involved as vascular cold sensors. Therefore, we aim to investigate the time-dependent changes in the vascular cold response during the onset of diabetes and to investigate cold-sensing behavioural changes as diabetes progresses. Due to the known damage that can occur to peripheral neurons in diabetes via hyperglycemia, we hypothesized that the vascular response to cold becomes dysfunctional. A dysfunctional constriction component is observed in the streptozotocin-induced diabetic mice. Using C57 Black 6 mice, we inject 50 milligrams per kilo of streptozotocin over 5 days daily. We then use a mouse model of localized cold. As you can see on the left-hand side, we use full-field laser perfusion imaging, which can record cutaneous vasculature blood flow in the hind paw of these mice. As you can see, these are two representative traces. So we record a 5-minute baseline recording, followed by cold treatment where we pause the recording. This is to allow the right hind paw of the mice to be placed into 10 degrees cold water for 5 minutes. Following cold treatment, we record the vascular response for 30 minutes. As you can see, in non-diabetic mice, there's a robust reduction in blood flow, which recovers to baseline over time. However, in diabetic mice, this response seems to be blunted at 2 and 5 weeks. When quantified, this becomes more evident. You can see the robust reduction in blood flow in a non-diabetic mice at 2 and 5 weeks. However, at 5 weeks, this is significantly reduced. Diabetic mice also become hypersensitive to innocuous cold at 5 weeks. Using a cold plate, we set the cold plate to the chosen temperature. We then record the time taken for the mice to initiate a nose-offensive response. This could be paw licking, paw jumping, or paw flicking. We use 30 degrees as our control, which is a non-cold temperature. We then use 20 degrees, which is innocuous cold. As you can see at 2 weeks, there seems to be no change in sensation. However, as the disease progresses at 5 weeks, we have an enhanced response to 20 degrees, which suggests hypersensitivity. To summarise and conclude, these results suggest to us that the sympathetic component of the TRPA1 dependent vascular response to cold is dysfunctional, which becomes evident as early as 2 weeks. Cold sensing behaviour, however, to innocuous cold becomes altered as the disease progresses, which was seen at 5 weeks. These results provide us with knowledge of the time-dependent onset of the neurovascular dysfunction in response to cold as diabetes impacts. Further study is currently ongoing to determine these implicated mechanisms of this impaired response. Thank you to everybody involved in my research, and thank you for listening.
My name is Christina Runela, and today I'm going to present the effect of NOx4 on cardiac glucose metabolism. The heart undergoes substantial remodeling in response to stress, which involves cardiac hypertrophy that can progress to heart failure. In recent years, it has been established that changes in cardiac metabolism are key in this process. Here, one important aspect is that stressed heart increases the use of glucose. However, the relationship between changes in substrate utilization and the functional state of the stressed heart remains unclear. We've been working with the redox enzyme NOx4 and found that overall, NOx4 is protective in a stressed heart via different mechanisms. Recently, one of these mechanisms has been identified as changes in intermediary metabolism and redirecting cardiac glucose away from oxidation. This prompts a question of the metabolic fate of glucose. Therefore, I established a methodology to determine the metabolic fate of glucose using a stable isotope labeled version of glucose. The metabolic intermediates originating from the labeled glucose will show an increase in mass with every incorporated carbon-13, which can be detected from mass spectrometry and is a readout of in vivo pathway activity. This is here illustrated as a comprehensive pathway map of glycolysis and its branch pathways and the TCA cycle, with kill plots indicating the isotopal abundances in yellow compared between NOx4 transgenic and wild-type hearts. NOx4 induces a complex series of changes. Here, I will just focus on one aspect, which is the Theorem biosynthesis pathway. This pathway stood out in particular due to the significant carbon-13 enrichment in the NOx4 transgenic hearts as compared to the wild-type hearts. This indicates that NOx4 overexpression increases the Theorem biosynthesis pathway activity. The metabolic flags results were also supported by Western blot and qPCR data, where we detected a significant increase in NOx4 transgenic mice of enzymes catalyzing Theorem biosynthesis, namely PHTDH, PSET1, and PSPH. Intriguingly, these changes were preserved in a model of chronic pressure overload and hence might be associated with the reported cardioprotective effects of NOx4. In summary, we show that NOx4 diverts glucose into the Theorem biosynthesis pathway, which was associated with an increased expression of its pathway regulating enzymes. These results indicate that NOx4 upregulates intermediary metabolism, which might produce nucleotides and amino acids integral for cell growth and survival. Thank you for your attention.
Hello and welcome to my 3-minute flash talk. My name is Simon Zhu and I'm a PhD student supervised by Professor Bijan Madurai. My project is on the role of CD34 positive cells in vein graft remodeling. In particular, I'm interested in the changes that vein grafts undergo when they are placed into arterial circulation. This is because vein grafts are still the most commonly used conduit in bypass surgery, but their problem has always been that of long-term patency. Studies have shown that vein graft patency after 5 years tends to be around 50 to 60%, despite our best efforts. Unlike in the arterial circulation, where the pathogenesis of atherosclerosis takes decades, vein grafts start undergoing this process in a matter of months to years after surgery. By looking at cadaveric grafts, we have known for many, many years that veins undergo a remodeling process when placed into the arterial circulation. Morphologically, they become more like to arteries with thicker and more muscular vessel walls. We've also established that the remodeling process is what contributes to their early disease. However, studies in this area have not yet determined the origin of cells that contribute to the remodeling process, and questions remain as to whether they are chiefly derived from vascular progenitor cells within the graft itself, or recipient-derived cells from either hematopoietic stem cell or local progenitor cell line. In order to address this, we have devised a lineage tracing strategy using an inducible knock-in mouse line. CD34 Cree-ER mice have been created that will express T tomatoes in cells that are CD34 positive after a course of tamoxifen. That means that all cells derived from CD34 positive lineages will remain red even after proliferation and differentiation. We then perform a vein graft model on these mice, whereby we place donor inferior vena cava as an interposition graft onto the common carotid artery of another mouse. By combining knock-in mice with wild-type grafts and vice versa, we can determine the relative contributions of vein-derived and recipient-derived cells in the near-intima of these vein grafts. We then perform histological analysis of vein graft specimens with in vitro studies of purified CD34 cell differentiation in order to determine the roles of host and donor stem cells in vein graft remodeling. This offers the prospect of new therapeutic targets for the prevention of vein graft restenosis due to adverse remodeling in the future.
So I'm going to hand the floor over now to Kevin and Suzette to kick off our second session of oral presentations.
Thanks very much, Alex. So we've got 3 great talks in this session, the first of which is Dr. Matthew Ryan, who's going to be talking about intracoronary physiological testing in the detection of myocardial viability and prediction of hibernation in ischemic cardiomyopathy. So over to you, Matt.
Thanks very much. Can you see my slides there?
Yes. Yes.
Perfect. So thank you very much for the invitation to speak. My name is Matthew Ryan, and I'm a third-year PhD student working for Professor De Barca Pereira on the St. Thomas's campus. So my subject area is ischemic cardiomyopathy, and put simply, this is left ventricular systolic dysfunction, which has been caused by coronary disease. Compared to other forms of dilated cardiomyopathy, it has a higher mortality. You have increased hospitalization and generally lower quality of life as a result of the fact that the causative etiology is coronary disease. And the particular thing about ischemic cardiomyopathy is compared to the non-ischemic cardiomyopathies, there are multiple pathophysiological mechanisms that lead to dysfunction. And clearly the common one that everybody will think about is generally infarction. However, the other key driver that's present in probably at least 50% of patients with ischemic cardiomyopathy is myocardial hibernation. And hibernation is the adaptive downregulation of myocardial function in the face of recurrent episodes of ischemia. The idea being that in order to obviate recurrent episodes of ischemia, the myocardium essentially does less to conserve the resource that's available in order to maintain myocyte viability. Over time, there is, however, a loss of contractile apparatus. There's changes in mitochondrial function and glucose utilization, and eventually glycogen deposition. And the key thing about this is if treated in early stage, hibernation is reversible. The myocardium can essentially be woken up again, but with time, that dysfunction can become irreversible. Why is this important from a clinical perspective? Well, it's key to decision-making, particularly has been considered around bypass surgery and stenting, the so-called revascularization procedures, with the idea being that if you revascularize and treat the coronary arteries to prevent these episodes of ischemia, that the myocardium can recover and return to normal. And that's been very elegantly demonstrated both in animal and clinical models. The problem from a clinical perspective is that to do this, you need 2 tests. You need to understand the coronary arteries to say, well, is there an ischemic substrate? Are there going to be recurrent episodes of inducible ischemia. And at the moment, that's probably best done with an invasive coronary angiogram as pictured on the left. And then you also need to do something called a viability study, which looks at the myocardium and determines whether or not there is viable tissue there and whether there is a potential substrate for hibernation that might be reversible. And again, there's multiple modalities with which you can do that, but probably at the moment, the clinical reference standard is what's called late gadolinium enhanced cardiac MRI, which is the image on the bottom right. And essentially that shows areas of black. So you can see at the bottom of the left ventricle there, that's black, thick myocardium, which is viable and alive. And then the areas of infarction as the other mechanism around the very bright white areas around the left ventricular apex. So my project was funded by the BHF and comprised of two primary studies. One, looking at coronary wave intensity analysis for the invasive detection of hibernation. And second, on improving the detection of hibernation with cardiac MRI. And that's based around two things. It's that if you're doing these two tests, would it not be better to be able to just do one test and then proceed directly? Because each of those two tests at the moment is only looking at one aspect of the pathophysiology of hibernation. And the question is, either invasively or non-invasively, can we actually look at both? So all the research procedures were conducted at St. Thomas's, at King's and at Bristol Royal Infirmary over March 2018 to April 2021. So the first was looking at coronary wave intensity analysis. And wave intensity analysis is seen as fairly novel, but it's actually a very simple concept. Quite simply, when you have left ventricular systole, you have contraction of the myocardium, and that drives a wave of energy back up the coronary from the distal microcirculatory bed back towards the aorta. And that's the BCW, the backwards compression wave that's seen there. And then as the myocardium relaxes in diastole, you get a backward expansion wave as the microcirculation expands, and a wave of energy is essentially sucked down the coronary from ostium to distal microvasculature. And you can measure that using something called a combo wire, measuring simultaneous pressure and flow in the coronary. And some very elegant work done previously by Calpa de Silva, again under Professor Pereira's supervision, demonstrated that in patients who've had an acute myocardial infarction, that those are good measures of viability. But the problem is that the acute coronary syndrome context has really fallen out of favor because the overwhelming evidence base is that these patients benefit from revascularization, whatever the state of the myocardium, rather than in the stable ischemic cardiomyopathy setting where viability is seen as more important. So we had a hypothesis that the backward compression wave and backward expansion wave energy might be non-inferior to late gadolinium enhanced cardiac MRI at predicting the presence of hibernation in ischemic cardiomyopathy. How do we do that? Well, we had a study where we performed a transthoracic echocardiogram at baseline to look at myocardial function. And then we did the current reference standard of cardiac MRI with late gadolinium enhancement. And then we also performed coronary wave intensity analysis in the cath lab during a diagnostic angiogram. We then followed the patients up as intended initially at 6 months with another transthoracic echocardiogram and did blinded analysis of baseline to follow-up echocardiography to determine whether there'd been an improvement in function. And that improvement in function is essentially the current reference standard for hibernation. So it became our primary endpoint. And we looked across the region, the whole territory subtended by the coronary artery that had been investigated, and looked at a reduction in wall motion score index greater than 0.5. Of 61 eligible patients, we included 40, from whom we had 61 coronary arteries with a mixture of different vessels, of which 41 territories were viable, determined by a late gadolinium enhanced threshold of 25%, and non-viable 20, and then had follow-up echocardiography available in 25 patients and 35 vessels, and that was primarily as a result of loss to follow-up due to COVID, as well as some patients who didn't want to attend again. The demographics were quite different to previous studies. This was a true ischemic cardiomyopathy cohort, so very impaired left ventricular systolic dysfunction, systolic function, very dilated ventricles, high NT-proBNPs, multi-vessel disease with high jeopardy scores. And so it is a true ischemic cardiomyopathy population. Of those territories, 25 territories were viable at baseline and 10 were non-viable. This is the ones we had follow-up echocardiography. And 17 territories recovered and therefore were determined to be hibernating. And 18 did not recover and therefore were determined to not be hibernating. And our data showed that there was a substantial difference in the backward compression wave energy of the recovering hibernating myocardium as compared to the non-hibernating, non-recovering myocardium. So the backward compression wave had a mean of about minus 5,500 and the non-recovering minus 1,900. Interesting, the backward expansion wave, which was the best predictor in the acute coronary syndrome context with Dr. De Silva's work, didn't differ. And if you looked at the areas under the curve, they were essentially comparable. So 0.81 for coronary wave intensity analysis and 0.77 for cardiac MRI with a p for difference of 0.65, indicating that they were roughly comparable as tests. And that's the scatter plot. So just looking at it in a little bit more detail. And so you can see in general, the orange dots are those who did not recover, the blue dots are those that did recover. And you can see generally, the lines are at the optimum threshold across of about 2,200 for wave intensity and at the clinical threshold of 25% for scar burden, indicating quite effective differentiating, although still quite an overlap down in the top left corner. And so it looked like coronary wave intensity was a good measure of myocardial hibernation and could be used in a clinical setting to expedite treatment. The second project was improving the detection of hibernation with cardiac MRI. So as I said, late gadolinium enhanced MRI is the current reference standard, but provides limited characterization of hibernation because it only assesses one part of the substrate. It shows that the myocardium is alive, but it doesn't show it's ischemic. You can look for ischemic myocardium with stress perfusion cardiac MRI. Again, King's has been really critical in the development of this. And stress perfusion is widely used to detect coronary artery disease in patients with normal LV function. And the seminal trial MR-INFORM was again run from King's, and you can see many members of the faculty were on that New England Journal paper. However, it's not been tested for this use in ischemic cardiomyopathy. So the hypothesis was that combining stress perfusion MRI and late gadolinium enhancement would more thoroughly and more completely characterize hibernation and may better predict the presence of hibernation determined through functional recovery again. We recruited 34 patients in this study, 29 completed follow-up, and that gave us 464 myocardial segments, of which 458 were dysfunctional. And this analysis was on a segmental rather than regional level, so at a slightly smaller level. That's using the AHA standard segmentation. Again, a very significant ischemic cardiomyopathy population, very high BC's coronary jeopardy scores, very dilated left ventricles, and very impaired left ventricular function. This fairly similar but standard protocol to previous is transvastatic cardiogram, cardiac MRI with late gadolinium enhancement, this time also with stress perfusion, and a primary endpoint of segmental functional recovery at 6 months with a reduction of wall motion score index greater than 1. Blinded analysis of the cardiac MRI by an expert reader, and viability and hibernation was predicted if there was a scar burden of less than 25% on late gadolinium enhancement alone, or less than 25% late gadolinium enhancement and ischemia on the cardiac MRI with the dual protocol. So for the standard protocol with just late gadolinium enhancement, it was very highly sensitive at 78%, but poorly specific with only 26.8% of segments deemed viable, actually demonstrating hibernation. Perfusion really actually showed the reverse. It didn't result in an overall improvement in accuracy, but sensitivity in this case was 34%, but specificity improved to 75%. Relating to a positive predictive value of 44% and a negative predictive value of 66%. So it appears that the addition of perfusion is giving you a far more specific test at a significant cost of sensitivity. We looked across a variety of factors, the presence of myocardial viability, determined just on the late gadolinium enhancement, perfusion defect presence, baseline wall motion score index, and revascularization, and actually found that it was viability, baseline wall motion score with worse wall motion being predictive of a greater recovery, and revascularization that were the best predictors, and perfusion defect wasn't an independent predictor. And looking at this, it appeared one of the reasons was that it is likely that perfusion scanning was underappreciating the burden and extent of coronary disease, at least when determined by a BC's coronary jeopardy score on the left. You can see the coronary jeopardy predicted something like a 90% ischemic burden, and perfusion reliably somewhere under 50%. So in conclusion, coronary wave intensity analysis does detect hibernation during cardiac catheterization and allows a single assessment of coronary and myocardial function in one procedure. Equally, stress perfusion MRI in a non-invasive manner provides a greater specificity, but at the cost of a reduction in sensitivity in detecting hibernation. And these techniques could be used to refine the identification of hibernation and better inform treatment decisions in ischemic cardiomyopathy. Future work for wave intensity analysis will involve working out real-time wave intensity analysis, and we're going to be doing that with Jack Lee and the heart and vessel modeling group from Imaging Sciences, the prospective validation of wave intensity using a multi-center study, and the integration of wave intensity with invasive arterial physiology like fractional flow reserve, which we'll do once we have the extra follow-up patients in. Stress perfusion MRI, the plan is to do quantitative perfusion with Professor Sherry Biri, as well as do a comparison of stress perfusion MRI with arterial physiology to try and understand a bit better that discrepancy in ischemic burden between the two modalities. I'd like to thank Professor Pereira, Professor Chiribiri, Professor Marber for the supervision, and all of the teams at Guy's and St Thomas's, KCH and Bristol Royal Infirmary, as well as the Catheter Lab teams, the patients, and of course the British Heart Foundation for their support during all of this. Thank you very much.
Fantastic. Thanks very much, Matt. Do we, perhaps just to stick to time, do we have any questions? Professor Shah?
Matt, quick question, really nice presentation. On the wave intensity, you showed that the compression wave is the main difference. And I was wondering whether you think that is just a reflection of increased contractile activity driven by dobutamine. In other words, I mean, devil's advocate, is this a very clever way of showing that a hibernating segment shows recovery of contractile function with low dose dobutamine?
So sorry, I should have clarified during. So as part of the protocol, we did do dobutamine wave intensity analysis because we had this hypothesis that it might work essentially like a stress echocardiogram. But actually, the data I've shown is the resting data. And actually, the dobutamine doesn't improve your, whether treated as a single marker or as an index of change, doesn't actually change anything. And if you look at the territories they are, if you look at both the MRIs and the echoes, as good as that is a measure of contractility, they're actually exactly matched in terms of resting wall motion. So it may be that hibernating myocardium, because there's more preservation of contractile apparatus compared to infarction, there may be something going on at a sort of lower and subclinical scale that we can't see on echo or MRI. But otherwise, it appears to be pretty matched from what we can see at a clinical level.
Okay, thank you very much, Matt, and I'll just pass on to Suzette to introduce the next speaker.
Great, thank you. So it gives me great pleasure to present the next speaker, Dr. Karim, and his topic on combining functional and anatomical imaging to improve limb salvage in peripheral vascular disease.
Ladies and gentlemen, thank you very much for giving me the opportunity to present my work today. My name is Lawan. I'm a trainee vascular surgeon and a second-year PhD student funded by the BRC. In the past 2 years, I've been working on developing a functional and anatomical imaging strategy for the ischemic limb. So critical limb ischemia is the most severe manifestation of peripheral arterial disease. Patients present with pain, tissue loss and gangrene as demonstrated here. Despite surgical intervention to improve the blood flow, which involves unblocking occluded vessels down to the foot, the outcomes are very poor. A third of patients will need an amputation within a year, another third will need re-intervention within a year. The dilemma the surgeon faces is how many of these blood vessels need to be unblocked to promote limb salvage. Below the knee, there are 3 vessels supplying the foot, and the traditional school of thought was that only one needs to be open to save the limb. Operating below the knee, however, increases the risk of complications by log fold. Currently, there's no objective way to measure success, and we are still very heavily reliant on the resolution of symptoms and signs. This can be subjective and can take up to 6 weeks to become clinically apparent. Our current imaging modalities, as demonstrated here, and geographies, only tell us about the patency of the large vessels and not the oxygenation of the muscles of the foot or the calf in which they supply, which is a more important determinant of limb salvage. We now know that vessel patency does not correlate to tissue oxygenation. So I'm going to give you an example of a 60-year-old gentleman, this one, who's got multiple comorbidities. You can see the large vessel in the pelvis is occluded and it's successfully stented, so anatomical patency has been achieved. Below the knee, a single vessel was patent. After the operation, the patient was noted to have a warm foot and the pain has slightly improved. The operation was deemed technically successful. Unfortunately, the patient came back 2 weeks later with a foot looking like this. We can see necrosis has extended, and unfortunately, we could not save the limb and the patient underwent an amputation. We want to develop an objective post-operative scan which measures oxygenation to identify limbs which remain poorly oxygenated to trigger more aggressive attempts at revascularization. So for this particular patient, it could be either more proximal work, or if the proximal inflow is okay, to tackle the vessels below the knee. So both the American Heart Association and the Global Vascular Guidelines have stated a need to develop a scan which measures oxygenation to improve the care we provide our patients with critical limb ischemia. They have deemed this a research priority to fill this evidence gap. So BOLD MRI, this is the blood oxygenation level dependent MRI. This is a non-contrast study which uses the paramagnetic properties of deoxygenated hemoglobin to assess oxygenation of the muscles. My department has done extensive work to develop a bold scan to measure oxygenation in the calf muscles of patients with critical limb ischemia. With the patient lying supine, a cuff is placed on the thigh and a coil is placed over the tibial tuberosity for reproducibility. Initially, anatomical T2 weighted images are acquired to delineate the anatomy. BOLD, which measures continuous T2 star over 3 phases of baseline, cuff inflation and cuff deflation. The images are then downloaded, and to analyze muscle oxygenation of a particular compartment, a region of interest is drawn around the muscle, and it's very important to include only the muscle and to exclude non-desirable structures like the bone and the vessels. T2 star signal intensity is then measured and plotted over time, and in a healthy volunteer of any of the muscle compartments, this is the pattern we expect. We see T2 star is dynamic and it changes with cuff inflation and deflation. At baseline, it's normal. When the cuff is inflated, we see a drop in T2 star, and when the cuff is deflated, we see an increase. The part of the curve that we're most interested in is this part here called the gradient. This is the rate of change of T2 star with cuff deflation, and it derives a single value of oxygenation for the patient. We've been able to show that T2 star is very sensitive to revascularization, where our ischemic patients, which are demonstrated in red here, have got flatter curves, and after revascularization, as seen in this purple line, the curve starts to resemble that of a healthy volunteer, as demonstrated in blue. We've been able to show that the gradient, as demonstrated here, has got good inter-scan and inter-user reproducibility, and that is a good measure of oxygenation for the calf, in that ischemic patients have got a lower mean gradient, and after revascularization, it significantly increases. So my work for the last 2 years has been to develop BOLD to make it a clinically useful tool. My hypothesis is the measurement of limb oxygenation using BOLD MRI will better guide the management of CLI patients and improve limb salvage rates. My aims to relate calf oxygenation to clinical outcomes. All the work we have done so far has been measuring calf oxygenation. I would like to extend that to start measuring the foot, the oxygenation of the foot, and I would like to relate that to both the calf and also clinical outcomes. To make it an objectively good, clinically useful tool, I want to enhance the speed, the accuracy and reproducibility of BOLD analysis by using deep learning techniques and artificial intelligence for scan analysis. So 71 patients were recruited and had pre-intervention BOLD scans, and there wasn't a significant difference when divided into Rutherford classification, age, gender and various comorbidities. When assessing calf oxygenation in CLI patients, the pre-intervention gradient was very good at assessing the oxygen deficit in the lower limb as the severity of the disease increases. As you can see, our CLI patients, which are our rest pain and tissue loss patients, have got a significantly lower gradient than our claudicants and volunteers. When a ROC curve analysis is performed, the pre-intervention gradient has high sensitivities and specificities, both of more than 91% in identifying patients who have CLI, and therefore it's a very good diagnostic tool. When calf oxygenation was related to clinical outcomes, we found that BOLD was good at predicting the success of revascularization. 35 patients were scanned post-operatively, and the success of surgery was defined as a resolution of pain, ulcer healing, no need for reintervention, and no major amputation. The fold gradient is the post-intervention gradient divided by the pre-intervention gradient, and we can see in the 12 patients who had unsuccessful surgery or poorer outcomes, we can see a lower fold gradient, significantly lower fold gradient compared to the successful group. When representing this in a ROC curve analysis, we can see that fold gradient has got high sensitivities of 83% and high specificities of 75% in predicting adverse outcomes after revascularization. So the foot is the most distal territory, is more susceptible to the damages of CLI as compared to the calf. Currently, there's no imaging modality which assesses oxygenation in the calf. Myself, I've been able to generate signals using the BOLD in the foot muscle, which are sensitive to revascularization. The mid-metatarsal point is taken as a reference point, and this here is the T2 weighted anatomical image, which is used for anatomical delineation. Below is the T2 star map of the BOLD, and you can see a region of interest drawn around the first interosseous muscle. As you can see, it generates T2 star signals which are very similar to that of a calf, in that ischemic patients have got flatter curves, as demonstrated in orange here, and after revascularization, the curve in grey starts to resemble that of a healthy volunteer. Again, similar to the calf, the foot gradient is very good at assessing the oxygen deficit in the lower limb, in that patients who have CLI, i.e. Breast pain and tissue loss patients, have got a significantly lower pre-intervention gradient compared to our healthy volunteers and our claudicants. I've been able to show that the foot gradient has good inter-scan and inter-user reproducibility, and that ischemic patients have got low gradients, and after revascularization is a significant increase. And for the 15 patients on individual plots, we can see in most of the cases there's been an increase in gradient with revascularization. So the current method is time consuming, it's laborious, and can take up to 4 to 6 hours to draw our regions of interest and analyze T2 star curves. Using deep learning and artificial intelligence techniques, we hope to automate the scan analysis to make it an objective and clinically useful tool. So, as you can see here, drawing the regions of interest can be very difficult, as it's very hard to differentiate between the different muscle compartments, and therefore it can be subjective. The deep learning techniques will be used to analyze 3D segmentations of T2 and T2 star images. These offer a higher signal to noise ratio, allowing better differentiation of the muscles from vessels, bone, and tissues and other non-desirable tissues. We have developed a data set by generating 3D segmentations of the calf and the foot of patients previously scanned. This is particularly important for the foot, which has a very small muscle bulk. As you can see from the 2D image here, it's very difficult to differentiate between the different muscles. The 3D segmentation will allow us to differentiate the anatomy much better, to give us a better assessment of oxygenation in the patients when drawing our regions of interest. So future work-wise, the whole point of this and our ultimate aim is to have bold MRI as part of a day-to-day workflow for treating patients with critical limb ischemia. I want to take this opportunity to thank my supervisors, Professor Bijan Modurai, Professor Alberto Smith, and Mr. Ashish Patel, and the rest of the department who I work with, and also everyone else who's involved in this research. Thank you very much for listening to me today. I welcome any questions.
Thank you very much. That's great. Does anybody have any questions? I guess then maybe I'll start with my question. So I understood from your talk that you were thinking of using this to kind of assess whether more aggressive surgical techniques need to be used to save limbs for these patients. But do you think this method could potentially be useful for catching pre-ischemic conditions? So for things like diabetes, where you might expect ischemia to develop, could you use this tool as a preventative measure before kind of clinically ischemia is classified?
Yes, you're right. The whole aim or purpose of the scan is to identify patients who need more work so we can intervene earlier. Most of my preliminary data has shown that it's very good at identifying patients who've got poorly oxygenated limbs. But however, it doesn't recognize ischemia before it has actually started and become clinically apparent. Because our healthy volunteers, so I'll go back to the slide. So when you're looking at the healthy volunteers, it doesn't make a differentiation between whether they have, are going to, develop ischemia or not. So no.
Thank you. Does anybody else have any other questions that they would like to ask? Okay, so I think then in the interest of time, we'll move on, but thank you very much.
Thank you.
Thanks very much, Suzette. So I now am delighted to introduce Adam Gwartz, who's going to discuss his work regarding venous end-stent stenosis in post-thrombotic syndrome. Over to you, Adam.
Well, I'd like to start off by thanking the organizing committee for giving me the opportunity to present my research work on the role of platelets in venous end-stent thrombosis. My name's Adam Gwartz. I'm a final year PhD student in Professor Alberto Smith's lab at St. Thomas's Hospital in the Academic Department of Vascular Surgery. So deep vein thrombosis affects 1 to 2% of the population, and it can lead to post-thrombotic syndrome when residual chronic venous obstruction or valvular reflux persists. PTS occurs in up to 50% of patients within 2 years of developing an iliofemoral DVT, and is associated with significant morbidity, including persistent pain, swelling, and ulceration. This chronic post-thrombotic patient group is the most challenging to treat, with patients having non-healing venous ulceration and poor quality of life. It's also been established that medical management alone is ineffective at treating post-thrombotic syndrome. Histological analysis from post-thrombotic tissue causing intraluminal venous obstruction demonstrates collagen and inflammatory cells. Venous stenting to overcome this venous outflow obstruction is associated with symptomatic improvement and has been proposed as a method to treat patients in whom conservative measures have failed. However, approximately 30% of patients will require reintervention, in some cases multiple times, for in-stent stenosis despite being on full therapeutic anticoagulation, and this is to prevent the original symptoms from recurring. Venous stent patency is likely influenced by a number of factors, including those associated with the patient and the operation. Kaplan-Meier analysis from our data demonstrates that reintervention is greatest in the immediate postoperative period, which can be seen here, as demonstrated in black, in a fall of primary patency, which describes maintaining stent patency without requiring reintervention. Additionally, venous disease-specific quality of life data also reinforces the importance of maintaining stent patency. The median vein squall score, the higher the better here, improved significantly postoperatively and was sustained at 24 months postoperatively in patients with patent stents. So taken together, we hypothesize that platelet activation contributes to venous in-stent thrombosis, and we aim to define the histological characteristics of in-stent thrombosis and examine platelet phenotype and function before and after venous stenting in patients who develop in-stent stenosis or have patent stents, and identify a specific platelet biomarker in chronic post-thrombotic patients. So to do this, specimens of in-stent thrombosis with and without nitinol stent material were analyzed to detect collagen and platelets. Flow cytometry was used to analyze platelet activation before and after stent placement. Patients were then prospectively followed up during ultrasound surveillance, and those with in-stent stenosis requiring reintervention were compared with patients with patent stents. Plate-based aggregation was used to evaluate platelet function in patients before and after stenting and in healthy donors. And an ELISA was used to detect soluble GP6 in pre-stent plasma samples of patients with chronic post-thrombotic syndrome and acute DVT. The schematic at the bottom demonstrates the timing of when the various samples were taken and of our ultrasound duplex surveillance program. So immunohistochemical analysis of acute in-stent thrombus 7 days post-stent insertion demonstrated a rich network of platelets, seen here in black. Analysis of chronic in-stent thrombosis revealed abundant collagen and platelets. Immunohistochemical analysis demonstrated both early and late forms of collagen deposition, collagen type 3 and type 1 respectively, reflecting the dynamic process of in-stent thrombus organization occurring over time. And in chronic in-stent thrombosis with stent material, the struts were surrounded by dense collagen. Collagen was the dominant tissue that was obliterating the lumen in these samples. So having identified the platelets in venous in-stent thrombosis specimens, we examined platelet phenotype before and after stenting in 45 case-matched patients. We looked at both platelet activation, here on the left, characterized by P-selectin exposure on the platelet surface, which occurs early following alpha-granule degranulation, and procoagulant platelets, characterized by both phosphatidylserine and P-selectin expression, seen here on the right. So pre and post-stent P-selectin exposure were compared between patients with in-stent stenosis requiring reintervention, seen here in black, with those who did not, seen here in white. So we observed a significant increase in P-selectin exposure pre-stent in patients who developed in-stent stenosis. No difference was observed post-stent. Figures on the right demonstrate a low percentage of procoagulant platelets in the circulation in post-thrombotic patients, and there was no difference observed before or after stenting. So next, we examined platelet function in post-thrombotic patients before and after stenting. We used an endpoint aggregation assay, which enabled us to simultaneously analyze multiple platelet agonists, which are listed on the x-axis. The y-axis represents a negative log scale of the concentration of agonist, which triggered a half maximal platelet aggregation response. In this figure, we can see that stenting did not affect platelet reactivity to any agonists used. We then examined pre-stent platelet reactivity in patients who developed in-stent stenosis, seen here in black, and those with patent stents, seen here in red. Platelet reactivity to collagen-related peptide, demonstrated here as CRP, is a GP-specific, GP6-specific platelet agonist, and it was significantly reduced in platelets who developed in-stent stenosis, which is denoted here by requiring an increased concentration of the CRP to achieve half-maximal aggregation. Next, looking exclusively at CRP, pre-stent platelet reactivity was significantly reduced in post-thrombotic patients compared with healthy controls. So next, since collagen-dependent platelet activation results in GP6 shedding from the platelet surface, we examined whether the observed reduction in platelet reactivity to CRP was due to GP6 shedding in the form of soluble GP6. Soluble GP6 was measured in pre-stent plasma samples of 30 patients with either acute DVT or chronic post-thrombotic syndrome. This figure demonstrates that chronic post-thrombotic patients had significantly greater levels of soluble GP6 compared with acute DVT patients. Looking exclusively at the chronic post-thrombotic syndrome patients now, this figure demonstrates significantly greater levels of soluble GP6 in in-stent stenosis, seen here in solid red, compared with patent stents, suggesting that the platelets of patients who develop in-stent stenosis have greater GP6-mediated activation pre-stent. So our results show that although in-stent thrombi are rich in platelets and collagen, venous stenting does not affect platelet activation or function. They do, however, show greater platelet activation prior to stenting in patients who develop in-stent stenosis, and these patients also have reduced GP6-mediated activation with loss of this integrin receptor from the platelet surface. We propose that measurement of pre-stent soluble GP6 may find utility in the risk stratification of patients undergoing deep venous reconstruction and help to predict which require closer surveillance. So when elective venous surgery restarts post-COVID, I'll be resuming my experiments examining thrombus formation on collagen under venous shear, which is a novel assay that we've developed in collaboration with a group at the University of Reading. Thrombus formation will be assessed in vitro using blood from post-thrombotic patients undergoing venous stenting and healthy donors in the presence and absence of platelet inhibitors using a microfluidic assay with chips that are coated with type 1 collagen, as this is the greater representation that we see. The level and the rate of platelet accumulation and in vitro thrombus stability will be of particular interest in these future experiments. Well, thank you very much, and I'd like to especially thank my supervisors, Professor Avilter Smith, Mr. Prakash Saha, and Professor Steve Black. Thank you very much for listening.
Fantastic. Thanks very much, Adam. Are there any questions? I've got a question, if that's okay. GP6 was increased in patients who went on to develop complications of stenting as such. Is GP6, is that independent of disease severity or quality of life scores pre-stenting anyway? Is it just a marker of severity?
So that's a good question. So GP6, the molecule itself, is in platelet worlds, it's a bit of a holy grail in that the thinking is that if you can inhibit GP6, you'll inhibit pathological thrombosis, but not affect hemostasis. So there's quite a lot of attention on it. Looking at disease severity, there is an association with an increased amount of collagen exposure in disease severity, because as you get reduction of inflow and increased thrombus burden from the post-thrombotic tissue, you get an increased amount of collagen exposure. But collagen in general looks at GP1B alpha and GP6 itself. So we don't see a necessarily increased amount of GP6 shedding in those that have reduced inflow, but we do in those people that go on to develop in-stent stenosis.
Okay, and in terms of the solution to this problem, is the answer purely more potent anti-platelet therapy like for example, is used in complex coronary disease?
So that's one of the hypotheses that we're working on. The idea being is that GP6 is unlikely to be targeted in our model now because it's existing at the point that we're actually intervening. So the thinking is that what we're exploring is whether the other inhibitors down the PTY12 and so on pathways are still intact, and the inhibitors that are established clinically for patients would work in this subgroup of patients that have both an increased amount of disease burden and also an increased amount of GP6 shedding.
Okay, well, thank you very much. Are there any other questions from the audience? I'll just check the chat. Okay, fantastic. Well, listen, thank you. Oh, we do have a question. We have a question from Orthi Kalwan Singh. Go ahead. I think you might be muted.
Okay, can you hear me now?
Yes, indeed.
Just a quick question regarding, was there any differences in patients receiving different antiplatelet therapies in your study?
So none of the patients had any antiplatelet therapy. So antiplatelets don't form a normal, aren't part of the normal anticoagulation protocol. So none of these patients typically get antiplatelets, or they don't at all preoperatively. As a protocol within St. Thomas's Hospital, we add an antiplatelet to those patients that require multiple reinterventions. But in the group at the start, when they have their first procedure, none have antiplatelets.
Thank you.
Okay, well, thank you very much, Adam, and thank you to the three speakers from this session. And that concludes this session. I'll pass over to Alex for the next flash talks.
Thank you very much, Kevin. Thank you for the chairs and the speakers of the oral presentations. I'm going to share my screen.
My name is Irrepressibility, and I'm a clinical research fellow in the Cardiac Arrhythmia Research Group. Atrial fibrillation, or AF, is the most common sustained heart rhythm abnormality, affecting over 33 million people. It causes the heart to beat irregularly, often faster than usual, and is associated with an increased risk of stroke, heart failure, and even death. Treatment of AF often involves a catheter ablation. Catheters are these long, thin tubes that are inserted into the heart, allowing us to map the anatomy and electrical conduction system within the heart. Specific catheters create these small focal burns, creating scar tissue that interrupts the electrical circuit. Now, AF requires two things to occur: a trigger to initiate it, and the mechanism or electrical circuit to sustain it. The trigger is additional heartbeats originating from these pulmonary veins here that feed into the left atrium. An AF catheter ablation in its simplest form allows us to electrically disconnect the veins from the atrium using continuous lesions, thereby eliminating the trigger. To date, our procedural outcomes remain modest at best, with between 30 to 50% of patients experiencing AF recurrence within 5 years, despite multiple procedures. So there is significant room for improvement. One of the main reasons for procedure failure is electrical reconnection of the veins with the atrium, particularly in areas where the atrial tissue is thicker, and this is where the problem lies. Too weak a burn and reconnection can occur, but too strong a burn can result in collateral damage to surrounding structures, such as the esophagus or the lung. The thickness of atrial tissue also varies between patients. Therefore, the optimal settings for creating lesions will also differ between individuals. Our study uses novel technology, allowing us to measure atrial wall thickness in real time inside the heart. We performed a pilot study in 4 pigs and collected 48 wall thickness measurements in the heart. These measurements were compared with pre-procedural wall measurements taken on CT, as seen in panel D. Panels A and B show no statistically significant difference was found between our technology and CT, whilst panel C demonstrates no systematic or proportional bias between modalities. Our novel technology allows us to take cardiac electrophysiology procedures to the next level, converting a previously two-dimensional structure that's wrapped into a shell into a four-dimensional structure that has thickness and form. It enables individualized optimization of ablation power whilst maintaining a low procedural complication rate from collateral damage. It has the potential to increase the number of patients who remain free of AF and reduce the number of procedures required to achieve this, optimizing patient care, but also reducing health economic costs of AF therapy. Thank you.
Hello everyone, my name is Richard Burns and today I'm presenting my investigation into the relationships between genotype and phenotype in cardiac shape. Cardiovascular disease is an increasingly prevalent issue in modern society, and cardiac shape has been established as a key risk factor for its pathogenesis, as well as an indicator of prognosis. Despite this, little work has gone into investigating the mechanisms that underlie cardiac shape, such as its genomic basis, which is the focus of this project. We have used newly available data from the UK Biobank to create the largest to-date cardiac atlas of 27,000 participants, which is soon expanding to 50,000, and are using this to perform novel genome-wide association studies into cardiac atlas-derived shape features using the UK Biobank's genome sequencing data. Machine learning-based cardiac biventricular contour data derived from the UK Biobank's CMR imaging study was used to construct diffeomorphic subdivision mesh models for the 27,000 participants at end diastole and end systole, the combination of which was then used to create a vectorized end diastole to end systole motion model. We derived statistical shape models from these through principal component analysis, summarizing modes of variation in cardiac shape across the cohort. Through visualizing these principal components, we can categorize the observed modes of variation into biological shape features. Z-scores per participant for each mode form the phenotypes of interest in our genomic analyses. As shown on the slide, the first 5 principal components of the end diastole atlas captured 72% of shape variation observed. These are some examples of the mode visualizations for the end diastole atlas. Cycling between plus and minus 2 standard deviations, we have principal component 1, which corresponds with the size of the heart, principal component 2, in which we can observe significant variation in the apical position and orientation. In our study design, we selected standard covariates for working with UK Biobank CMR data: height, age, sex, array type, adjusted value for systolic blood pressure, resting heart rate, and BMI. Participants were also excluded from our analysis based on having a non-European ancestry, a significantly low ejection fraction, having a significantly deviated Z-score from the mean, missing covariate data, or a previous major adverse cardiovascular event. Genetic QC for this genome analysis was performed with Plink with the filters mentioned below. In this study, the heritability analysis was performed with Bolt-Rimmel. Bolt-Rimmel estimates the proportion of variation in a target phenotype explained by genomic variation rather than environmental variation, and we found that for all 5 principal components, they had a significant heritability. This suggests that all 5 principal components are good targets for GWAS. Genome-wide association studies in this investigation were performed with Bolt-LMM and are still ongoing. A genome-wide association study assesses how loci in the genome correlate with a target phenotype. The Manhattan plot on the slide gives a summary of the results of principal component 1's genome-wide association study, with chromosomal location plotted on the x-axis and correlation with the phenotype of interest plotted on the y. We can observe several loci exceeding the boundary set for genome-wide significance and further loci above the threshold for borderline significance. These would all be targets for further investigation into their biological relationship with principal component 1. I want to say a massive thank you to my supervisors and everyone that's helped out with the project so far, and I'm open to questions. Thank you very much.
Hello everyone, I am Sofia Monaci from the Biomedical Engineering Department, and today I will be talking about a sub-project of my PhD, which focuses on automating the localization of focal ventricular tachycardia from information stored in implanted devices. Focal ventricular tachycardia is a life-threatening arrhythmia which is driven by early and repetitive firing from regions other than the sinoatrial node, as we can see in this picture. And the radiofrequency ablation is the most effective treatment, which consists of catheterizing ablating those sources. However, its success is dependent upon an accurate localization of these sources, and therefore the procedure is time-consuming, invasive, and it requires VC induction. To improve it, we aim to automate the localization of these focal VT sources by combining fast cardiac models with convolutional neural networks following a previous study. But instead of using ECGs, we intend to use the electrograms, EGMs, stored in implanted devices, in general named CIDs. The reason for this is because most of the patients already have implanted devices inside when they go to the cath labs, and the information stored in those devices and the information carried by the EGMs could be really useful in improving and speeding up the ablation procedure. We can see here the workflow of our study, which consists of simulating cardiac focal VTs in a 3D computational model. And that it is then used to generate electrograms from a modeled and generic implanted device. And the 16 different combinations of EGM vectors were then used as input to two CNN architectures that can return the location of a focal VT. And these locations can be compared, as we see here, in a 17-segment 2D model of the left ventricle that can be easily interpretable by clinicians, where we can compare the CNN outputs with as circles with the focal origins and diamonds, and we can see the close match between the two. With this study, we were therefore able to successfully automate the localization of focal VT sources from EGMs using and combining computational models with CNNs, and the mean localization was around 8 millimeter between the CNN outputs and the VT origins, which was a really good result considering that the average lesion size is roughly 8.5 millimeter and can go up to 12 millimeter. If you want to know more, feel free to ask me questions. Thank you very much.
Okay, welcome back everyone. So we've reached now the final and third session of our 10-minute oral presentations. Without further ado, can I please ask Jaina and Carlo to come on board to chair the last session, please?
Good afternoon, everybody. Hi.
Go for it, Carlo.
Okay.
Yep.
Good afternoon and welcome to oral presentation session 3. Today I'm going to introduce you to Miss Ella Reed, which is a final year PhD student. And Ella got her degree in chemistry at the University of Bath. Today she is going to talk about the development and the application of engineered vascular tissues. And over to you, Ella.
Great, thank you. So good afternoon, my name is Ella Reed and I'm a third year PhD student over at the James Black Centre in Denmark Hill. Today I'm going to be talking about a part of my PhD project, which has been on the development, characterization and applications of primary aortic smooth muscle cell engineered vascular tissues. So smooth muscle cells exist in the arterial wall surrounded by a functional extracellular matrix, or ECM. They're a particularly interesting cell type for their ability to switch their phenotype from classical phenotypes such as synthetic and contractile, in addition to other phenotypes such as osteogenic or pro-inflammatory in response to vascular injury or environmental cues. In line with this, recent technological advances in cell lineage tracing and single-cell RNA sequencing has revealed that there are multiple different subsets of smooth muscle cells present in atherosclerotic lesions. These different smooth muscle cell subsets can be characterized by altered cell marker expression and ECM protein secretion. And indeed, changes in smooth muscle cell phenotype alongside ECM remodeling can contribute to the development and exacerbation of arterial diseases, including atherosclerosis. Currently, laboratory researchers utilize two-dimensional cell culture models extensively to study smooth muscle cells and their secreted ECM in vitro. And although incredibly useful, 2D cell culture is intrinsically limited by not being representative of the in vivo cell environment. For example, there is non-physiological stiffness of tissue culture plastic, a lack of diffusion gradients, a lack of 3D cell-cell and cell ECM interactions, in addition to reduced expression of mechanically regulated proteins such as proteoglycans. So to overcome the limitations of 2D cell culture models, researchers have developed novel 3D models. One of these models include fibrin-based engineered heart tissues, and our collaborators in Hamburg who developed this model show that iPSC-derived cardiomyocyte cells within this 3D tissue environment beat spontaneously and, importantly, give more physiological readout than 2D cultured cells. My project has involved adapting this model for the three-dimensional culture of primary aortic smooth muscle cells by making engineered vascular tissues, or EVTs. So how do we make engineered vascular tissues? So EVTs are formed by utilizing an agarose mold and a PDMS rack. A smooth muscle cell fibrinogen mask mix is then pipetted into these molds. On addition of thrombin, fibrin polymers rapidly form, allowing for the smooth muscle cells to be encapsulated. After approximately 1 hour at 37 degrees, the EVTs have set, and we can remove them from the agarose molds and place them into fresh media for prolonged culture. So as already mentioned, in vivo smooth muscle cells are surrounded by a functional ECM, consisting of arterial collagens, proteoglycans, and also elastin. Longitudinal sectioning of EVTs made from primary mouse smooth muscle cells and consequent immunostaining has shown the alignment of cells and accumulation of ECM proteins within the EVTs over time in culture. In particular, a proteoglycan and a collagen stain show accumulation of these ECM proteins over time. So given that we show accumulation of ECM within EVTs by immunohistochemistry, we next wanted to explore exactly what ECM proteins are accumulated using untargeted mass spectrometry. Here we took 3 different biological replicates and 2 technical of murine EVTs and control 2D cultured smooth muscle cells and harvested them at day 1, day 7, and day 14 to investigate their ECM expression profiles at the different time points. So this heat map shows some of the ECM protein changes within the EVTs at day 1 and day 7 of culture. At day 7, we see the upregulation of proteoglycans such as Verskin, alongside calcium-binding proteins such as matrix GAR protein. Molecular pathways that are significantly upregulated in EVTs at day 7 therefore include ECM structural proteins alongside calcium ion-binding proteins. And given the upregulation of calcium-binding proteins, I went on to investigate whether EVTs could be utilized to study vascular diseases such as calcification in vitro. Here you can see that under calcifying conditions, smooth muscle cell calcification is initiated at the day 7 time point. And we want to study this further to compare mechanisms of calcification in traditional 2D cultures compared to in EVTs. So when we then went to look at ECM remodeling within EVTs at the later day 14 time point, we can see an additional increase in ECM protein secretion. Going from day 7 to day 14, the upregulated pathways include ECM organization, system development, and also cell matrix adhesions. And interestingly, when I looked at differences in ECM accumulation in EVTs compared to 2D cultured smooth muscle cells at this time point, the most upregulated proteins in EVTs included collagen 4 family proteins and the proteoglycans biglycan and Verskin. And Verskin is of particular interest to us for its role in arterial health and disease. So first of all, what is Verskin? Verskin is a large aggregating proteoglycan that in its monomeric form consists of a core protein covalently decorated with sugar molecules called glycosaminoglycans. Through an interaction with hyaluronic acid molecules facilitated by its link protein hapten-1, Verskin can form large intermolecular aggregates and thus can contribute to the mechanical and biochemical environment that smooth muscle cells experience in vivo. And Verskin is readily processed by ADAMTS family enzymes. Our laboratory has shown an importance of ADAMTS-mediated Verskin turnover in different arterial pathologies, including aortic aneurysm and also atherosclerosis. So to study enzyme-substrate relationships in mouse models, mouse models are commonly used. And in our lab, for example, we have a mouse model whereby the protease ADAMTS5 is mutated and non-functional. In an ex vivo organ culture model, we show accumulation of ECM proteins, including Verskin and its linked protein hapten1 in the ADAMTS5 mutant aortas compared to control aortas. And additionally, we show a compensatory upregulation of the other ADAMTS family member, ADAMTS1. So as Verskin accumulates in EVTs, but not in 2D cultured smooth muscle cells, we decided to seed smooth muscle cells isolated from wild type and ADAMTS5 mutant mice into EVTs and study their ECM. By using an antibody that specifically targets ADAMTS cleaved Verskin, we demonstrate how in wild type EVTs, Verskin is readily being cleaved by ADAMTS5, whereas in ADAMTS5 mutant EVTs, we see significantly reduced Verskin cleavage. This therefore demonstrates that our EVT model may be a superior in vitro model to study ECM turnover in smooth muscle cells from different genotypes. Finally, I want to discuss the application of EVTs for pharmacological studies. As TGF-beta signaling is an important mechanism in regulating the smooth muscle cell contractile phenotype and also matrix synthesis, we wanted to investigate the contribution of TGF-beta signaling on EVT remodeling. To this end, we cultured EVTs under control conditions in the presence of TGF-beta and also in the presence of a TGF-beta receptor inhibitor that essentially blocks downstream TGF-beta signaling. Interestingly, we show that EVTs contract over time in culture under control conditions. However, TGF-beta supplementation massively increases this, with significant shortening and fattening of the construct occurring. In contrast, blocking TGF-beta signaling leads to an opposite effect, and lengthening and thinning of the tissue construct, and essentially loss of EVT integrity. I'm currently in the process of further characterizing smooth muscle cell phenotype and ECM secretion in EVTs under these conditions. So in conclusion, EVTs have been formed from primary aortic smooth muscle cells, and using a variety of techniques, I've demonstrated that cells within this model are both viable and secretory. Furthermore, ECM accumulation after prolonged culture is different in EVTs compared to 2D cultured cells. Finally, I've demonstrated that EVTs can be used for disease modeling, such as in the case of vascular calcification, for genetic studies whereby smooth muscle cells from different genotypes can be seeded within EVTs and their ECM studied, and for pharmacological studies, such as for studying TGF-beta signaling. In summary, hopefully I've demonstrated that EVTs may be a better in vitro model to study smooth muscle cells and their ECM in the context of arterial diseases. Finally, thank you to everyone in my group who has supported me in this project, in particular my supervisor, Professor Manuel Meyer, and also Dr. Adam Fellows, who helped to establish this technique in our lab. Thank you to our collaborators and also, of course, the British Heart Foundation for funding this project. And thank you for the opportunity to speak today.
Thank you very much, Ella, for this nice talk, very interesting. And it is now the time for some question. Does anybody has a question? Hannah.
Oh, sorry, I can't hear you.
Hannah, I can't hear you. You have to switch on your microphone.
Oh my God.
Yeah.
Okay, I'm so sorry. Yeah. So first of all, good afternoon. Really nice talk. I really like your in vitro model, the EVT. I think it's a really nice and novel model. You mentioned, of course, that you can actually extend it to different disease models. Totally fine. Like, for example, you mentioned vascular calcification, but have you thought about other possibilities? So have you thought about other models where you can extend such a, because it's a really broad in vitro model that can be extended somewhere? Have you thought about it?
Yeah, exactly. So at the moment, I'm just trying to characterize the model at baseline and get as many readouts as possible to understand the phenotype and also the ECM secretion within the model. And then I'm moving on to look at vascular calcification alongside other measurements as well. So I think the model's really diverse and there is a lot that we can do there. But yeah, it's a case of getting really sort of thorough and definitive readouts for this model, I think. So it's a lot of biological and technical replicates at the moment. But I think you look at a lot more things.
Indeed, yeah, technically it should be complicated, at least. Yeah, okay, thank you, a really nice talk.
Thank you.
Thank you for this question. Are there any other questions? I can't see anything. Just a question for you, Ella. Have you tried to co-culture your model with endothelial cells?
Yeah, so of course, smooth muscle cells and endothelial cells form really important interactions, and it is something that hopefully eventually we will look at. So again, with the other sort of disease models, I think this is something that once the model's more established, we can add endothelial cells and look at creating a co-culture model out of this as well, which would be really interesting.
Okay, thank you very much. And I think we are fine with this, and we can go to the next talk.
Perfect.
Thank you, Ella. Oh, yeah, Jaina, I pass this to you now, and yeah, that's it.
Thank you, Carlo. I'll be presenting our next, introducing our next speaker, Mihai Prună. He'll be presenting his work on the effect of beta-adrenergic signaling on intercalated disc architecture. So, Mihai, if you're there, you could start to share your screen.
Hello. Thank you.
Okay.
Share screen. Can you see the presentation?
Yes, but you need to make it into, yeah, perfect.
Okay, great. So today I'm going to talk about the work I've been carrying out in my PhD, which is the effect of beta-adrenergic stimulation on intercalated disc structure. The intercalated disc is a specialized structure in the heart which ensures a mechanical and electrical coupling between neighboring cells, and it was originally classified as consisting of three types of cell-cell contacts: adherent junctions in blue, desmosomes in pink, and gap junctions in green. But more recent work has shown that there's a lot of crosstalk between these elements, particularly in the adult mammalian heart. So why it is important to study the structure of the intercalated disc? It's because its architecture is affected in dilated cardiomyopathy, which is a disease of the cardiac muscle, in which the ventricle wall becomes much thinner, and also there is systolic dysfunction. And the first insight into the architecture of the intercalated disc came from studying the cardiac cytoarchitecture in NLP knockout mice, a model for DCM. So in these mice, the intercalated disc appears more convoluted, as seen in by electron microscopy. And in terms of protein levels, the expression levels of N-cadherin, plakoglobin, which are adherent junctions protein, are upregulated, and the levels of connexin 43, which is a gap junctions protein, appear to be slightly downregulated. Another disease where the intercalated disc is affected is arithmogenic capture. This particular cardiomyopathy, unlike dilated cardiomyopathy, there are some other histological changes in the myocardium, particularly fibrofatty replacement of cardiac cells. And this was initially believed to occur only in the right ventricle, but now it is accepted that also the left ventricle is involved. And in patients with the disease, if we're taking a look at the ultrastructural level, we could see that in healthy patients, the desmosome appears as a thick plaque at the intercalated disc, but in samples from patients with ACM, this is dissolved and the desmosome appear as individual spots. And arithmogenic cardiomyopathy has been defined to be a disease of the desmosome because over half of mutations in ACM are found in proteins of the desmosome, including plakophilin-2, desmoplakin, desmoglein-2, desmocolin-2, and plakoglobin. And here I've included their location in the protein. And the aim of my project was to look at the architecture of the intercalated disc in the presence of beta-adrenergic signaling. And beta-adrenergic signaling is overstimulated in patients with heart failure because the failing heart is characterized by overactivation of the sympathetic nervous system, and this results in increased levels of catecholamines. In acute beta-adrenergic signaling, this triggers adaptive changes in the myocardium, such as increase in heart rate, contraction, and relaxation, which are initially beneficial because they maintain cardiac output. But sustained long-term beta-adrenergic signaling results in harmful effects on cardiomyocytes, including pathological hypertrophy, cell death, and arrhythmia. And because the heart is bombarded with these high levels of catecholamines, the heart is also responding in turn with a decreased number of beta-1 adrenergic receptors. And this is believed to be a protective mechanism. And also the fact that the downregulation of beta-1 receptors is protective goes hand in hand with the use of beta blockers in the treatment of heart failure. And multiple clinical studies have been carried out showing decreased mortality, cumulative mortality in patients given a beta blocker. In this case, I've included a graph from a clinical study carried out in 1999, Merit-HF. And in this study, patients were given metoprolol or placebo, and in the presence of metoprolol, the cumulative mortality was decreased. And later on, there was a sub-study which showed that this decrease in mortality, particularly from preventing worsening of heart failure, was due to remodeling of the myocardium. And as shown here, there was a decrease in left ventricular and diastolic volume, and the ejection fraction was increased in patients given the beta blocker, even after 6 months of treatment. And something which added more to this background was the discovery that the beta-1 adrenergic receptor in the mouse heart is mostly localized at the intercalated disc. And further studies show that treating cardiomyocytes, in this case, they used a cell line, which was L1 cell line, treating it with beta-adrenergic agonist, isoprotenolol, triggers changes in the expression of desmoglein-2 at the intercalated disc. So in both confocal images and in images acquired by electron microscopy, there was the desmoglein signal appeared much longer at the intercalated disc. And they also carried out some dynamics experiments, some FRAP experiments. So they expressed desmoglein-2 tagged to a GFP in this cell line, and they monitored their dynamics. And in the presence of forskolin rolipram, which activates cyclic AMP signaling, there was an increase in mobility. So this suggests that beta-adrenergic stimulation, it's also affecting cell-cell contacts. And this term was called positive adhesiotropy. And this means that there's an increase in intercellular cohesion between cells. So the aim of my project was, because the previous work was carried out in a cell line, which is an atrial cell line, and the intercalated discs are not properly matured enough. I'm looking at cardiac cytoarchitecture, the intercalated disc in primary cells. So in my PhD, I've been using neonatal rats, cardiomyocytes. So we've been taking their hearts, and then we're looking at their cytoarchitecture. Throughout my PhD, this was not an easy task because you were working with primary cells. You could get bacterial and fungal contamination. You could get increased proliferation of fibroblasts. You could get over-digested cardiomyocytes that do not spread on substrate. So overall, only 50 or 60% of prep were usable. And on top of that, I'm looking only at cardiomyocytes who have a neighbor because I'm looking at cell-cell contacts, and this made things even more challenging. But even so, I went on, and what was done before in the lab was treating neonatal rat cardiomyocytes with isoprotenolol. results in massive arrhythmias in non-altered cardiomyocytes. And from this, I started to look at the architecture of the intercalated disc in the presence of beta-adrenergic stimulation. And the first protein was to look was the desmoglein-2, because this was also shown in the previous paper that it's affected by adrenergic signaling. So I thought this would be a good start. And I started different time course experiment, different hours, to see how is isoproterenol affecting the junctions. So to my surprise, at earlier time points, 1 hour and 6 hours, desmoglein-2 was not yet found at the intercalated disc, and it was found in cytosolic complexes. And isoproterenol treatment seemed not to affect much its localization. Only at later time points, after 1 day of treatment, desmoglein-2 was finally localized at the intercalated disc. And when I quantified the signal width of this desmoglein-2 signal, I've seen no significant change in its width. And I also look at the protein levels, and again, didn't seem to be massive change in protein levels either after 1 day. So then the next protein I looked at was plakoglobin. And I chose this protein because in the study was also shown that plakoglobin phosphorylation on serine 665 could mediate this change and this reorganization of the intercalated disc, because when they expressed constructs bearing a mutation from serine 665 to alanine, this reorganization is prevented. So this early experiment suggested that it could be this phosphorylation that perhaps triggers a conformational change in plakoglobin that could affect the reorganization. And then later work, more recent work, shown this to be true in a model, in a mouse, and they developed an antibody against this particular residue, and indeed they found an increase in its phosphorylation. And I also look at plakoglobin following the same time course, and at earlier time points, plakoglobin, unlike desmoglein-2, is found at the junctions, but doesn't appear to be a significant change in the appearance of the junctions. But what I found to change was only at 1 day of treatment, and whereas in control, the junctions appear more linear. And in the presence of isoproterenol, I've seen this increase interdigitation or formation of the spiky junctions. And when I quantify this, I could see an increase in the number of spikes in the presence of isoproterenol. And this phenomenon was seen also in other cells, and in mouse keratinocytes and in endothelial cells, and they are usually associated with stronger junctions. In terms of expression levels, these don't appear to change so much, so it could be that it's a redistribution of protein. And because plakoglobin is a promiscuous protein which associates both with adherent junctions and with desmosomes, I also look at proteins more particular for each of these two complexes. So first I look at plakophilin-2. So plakophilin-2, like plakoglobin, was found at the junctions at earlier time points. And when I looked at 1 day, I did not see a significant change in the number of spikes like I've seen with plakoglobin. And in terms of expression levels, this don't appear to change either. And then I look at the protein associated with adherent junctions classically, so beta-catenin. And I did the same time course experiment looking at 1 hour, 6 hours, and 1 day. And at 1 day of treatment, I've seen an increase in the number of spikes. So again, the junctions appear to be more diluted. And then I look at the expression levels. These don't appear to change either, so it's more likely that it's a redistribution of protein. I look at the expression levels of other adherent junctions, and these don't appear to change either. A summary of what I've done so far, and because only proteins affected in associated with the hearing junctions seem to be changing and seem to be causing this convolution in the intercalated disc. We thought that this might be caused by actomyosin, and I took a look at the myosin light chain 2 isoform expressed at the intercalated disc, and when this is closely associated with proteins of the intercalated disc, because when alpha-catenin was deleted and a double knockout was produced, this protein was no longer found at the intercalated disc, but was found randomly distributed throughout the cell. So I thought this would be a good marker to see whether actomyosin machinery could be involved, or whether there's any change in its phosphorylation levels. And also in the MLP knockout mouse I presented earlier, these levels appear to be upregulated again. So even more evidence, which made me look at it in this model system. And when I use this antibody in the neonatal red cardiomyocytes, I've seen that in the control, this phosphomyosin light chain appears as little dots across the membrane. And when isoproterenol was given to cells, this signal appeared to be along the spikes of the cell-cell contact. So and when I quantify this, it seemed to be an increase in phosphorylation. So this suggests that myosin could be involved in this junction remodeling at the intercalated disc. And then I tried an antibody I tried the drug to see whether blocking myosin activity can affect the intercalated disc structure. So I used blebistatin, because this was also used in other cell types. And when I treated the cells with blebistatin, I could see that in phenylephrine and isoproterenol, I saw the spikes forming, but when I give the cells blebistatin for 30 minutes, I could see that the junctions return to a more linear state. So this suggests that it's definitely a contractile response, this junction remodeling. And because blebistatin is toxic to the cells, I also took a look at the sarcomeric architecture and in the presence of blebistatin, and the architecture appears to be intact in these cells after 30 minutes of treatment. So summary so far, beta-adrenergic stimulation results in thicker junctions as observed by staining with antibodies for components of adherent junctions, beta-catenin and plakoglobin. These changes start day one after isoproterenol treatment, and this also coincides with the time desmoglein 2 reaches the plasma membrane. Beta-adrenergic stimulation increases the phosphorylation of threonine 18, serine 19 on non-cardiac myosin light chain 2, and the phosphomyosin light chain decorates the JAG membrane, suggesting that it could play a role in its formation. And blocking myosin 2 activity with blebistatin prevents spike formation. So this suggests overall that actomyosin machinery could be involved in junction remodeling. So acknowledgement, and thank you British Heart Foundation for the funding and you for listening today. Thank you.
Thank you, Mihai. That was a really great talk. So we're going to take some questions. Anyone got any questions? Yes, Anna.
Hi. Hello.
Hello.
So, yeah, first of all, a really nice talk. You did a great job, descriptive, quite thorough job, so really nice. So my question is quite broad. It's more like, how do you see your project, like in the short, middle term? Like, in which direction you would like to actually develop it from now onwards?
From now onwards. So I think this is important because it characterizes the changes which occur in the presence of isoproterenol treatment, and because it's also associated with arrhythmias, it could be a model to study intercalated disc proteins. So for example, there are so many mutations in desmoglein 2, plakoglobin, plakophilin, all the other desmosomal proteins which have not been completely studied. So I think would be a good idea to look at to create adenoviruses to express them in the cell and look at their dynamics between these different mutations. Also, the beta-adrenergic receptor. So it was shown that it's the intercalated disc, but the antibodies I was using, they were not that great. So probably a good idea would also be to take a look at to create an adenovirus for the beta-1 adrenergic receptor and see where that localizes and whether isoproterenol changes its localization, or it will also be interesting when in this model is beta-1 adrenergic receptor downregulated. So there are lots of questions which are still unanswered.
Okay, yeah, really nice. I mean, I can see that you have a really broad and a lot of ideas, and that's always nice, and that's what we're always hoping. Thank you.
Thank you.
Okay, great. Thank you, Mihai. I'll pass it back to Alex in the interest of time, and he'll present our guest speaker. Thank you.
Brilliant. So thank you all for your presentations. That concludes the end of the oral presentations and flash talks. Okay, so we're running a little bit over. So this is the moment in which I invite Anna to give her talk. And just to sort of run off very quickly Anna's biography. Anna was born in Algeciras in Cadiz, in Spain. She moved to the University of Seville to do her undergraduate degree in biotechnology. She then went to Madrid to study an MSc in neuropsychopharmacology. And her interests in biomedical research then led her to the Netherlands, where she did a PhD in Maastricht University in the Department of Pharmacology and Personalized Medicine, where she completed her PhD in 2018 with a cum laude. She then stayed on to do some postdoctoral work there, and recently became Deputy Head of Research in the same department, and was granted a Koostra Talented Fellowship. And now, currently, she holds a joint post. So she's also based at the University Hospital Essen as well. And I think she's taking this call from Maastricht, but I could be wrong. Okay.
Indeed, yeah, definitely.
Okay, so Anna, thank you so much. It's been a long time since the first time we invited you. Because we wanted you to speak at our 2020 symposium, and unfortunately, because of COVID-19, that wasn't possible. But we're really pleased to have you here, and so happy that you're able to make it this year as well. So I'm going to hand over the floor to you now, and if you could share your screen, that would be fantastic.
Yeah, perfect. Thank you. Okay, let's hope. Okay, do you see my screen?
Yeah, that's perfect.
Okay, perfect. Okay, so first of all, good afternoon, and I want to really thank the organizers for giving me the opportunity to be here today presenting our work. It's definitely a pleasure. So my talk is indeed divided into two main parts. So I'm going to start talking about breathing ischemia, about stroke. But why? Why does it matter? Why stroke? Well, stroke is at the moment the 5th cause of death and 1st cause of disability worldwide. At the moment, there is only one drug available, RTPA, with more than 30 side effects. So that means that only 20% of the patients, or even less, receive treatment at the end when they reach the clinics. And even more important, there is no neuroprotective therapy available right now. In fact, more than 1,000 experimental treatments have been tested for stroke in the last 20-30 years, and what was the success rate? Actually, none. Okay, nothing worked. At the moment, we have no neuroprotective therapy for stroke. And that's why almost all pharma companies have completely withdrawn from stroke research, because it's costly, it's tough, and not really going anywhere, at least in the last years. Now I want to focus and talk about reactive oxygen species. They are known to be part of several physiological roles like angiogenesis, blood pressure regulation. However, they are also been suggested to be part of several pathomechanisms, okay, including stroke. So our approach when we started with this projects, it was to inhibit the main source of ROS using specific drugs. And of course, NADPH oxidases are one of the major ROS sources. So in the beginning, we started validating NOx as a possible target for stroke using an in vivo stroke animal model. So here what we do is to occlude the middle cerebral artery. And we found, using knockout animals, that neither NOx1 or NOx2 were playing a role. However, NOx4 knockout animals presented less infarct size. But we wanted to go one step forward and know where exactly NOx4 was playing a role. And for that, we generated cell-specific NOx4 knockout mice. So we generated endothelial, NOx4 knockout, smooth muscle cell, and neuronal. And we saw a reduction in infarct size in endothelial knockouts, neuronal knockouts, and nothing in the smooth muscle cell. Also together with some neuromotor functioning. So at this point, we know where the problem is happening, right? So the question is how? So for that, we found that the neuronal NOx4 knockout animals, they had a significantly reduced apoptosis level, as you can see here, while the endothelial NOx4 knockout presented a vascular phenotype with a reduction of blood-brain barrier leakage. But here, in all this story, we should not forget that we have another player, NOx5. NADPH oxidase 5 is not expressed in rodents. So we actually had to generate a humanized mice model, inserting the NOx5 gene in the mouse. And then using this in vivo stroke model, we identified that these animals have increased blood-brain barrier leakage, increased ROS formation, and also increased infarct volume. So we can actually say that these animals, and therefore NOx5, is detrimental for stroke. But we wanted to see when. So when NOx5 was actually modulating ROS and how that was happening. So for that, we moved to an in vitro animal model where we used hippocampal brain slices. And here we saw that NOx4 was upregulated around 4 to 6 hours after reperfusion, while the role of NOx5 was indeed more earlier in the reperfusion period. So it was about 30 minutes until 1 hour after reperfusion. So we can see here in the course that NOx5 is playing a role really early after removing the occlusion, while NOx4 it's a little bit later after the reperfusion. And this can be essential for a neuroprotective therapy post-reperfusion. So we can actually combine the removal of the clot in the brain, something that can be done using thrombectomy, with the administration of a compound, just at the same time to avoid any post-reperfusion injury. However, I already mentioned that single treatments in stroke, they have been failing during the last 20, 30 years, okay, that we don't have too much. And we didn't want to do the same again. We didn't want to make the same mistake. So in relation to that, we need to keep in mind that in current therapies, it is common to first identify different symptoms, right? Then these symptoms are, of course, related to different targets. And then we treat them using 1, 2, or 3 unrelated drugs. And that is what we could consider classical combination therapy. But here, I'm proposing a different strategy, network pharmacology, where we focus on the causal mechanism underlying the disease. So in this case, I will target a complete disease model, of course, including different targets, and we will treat them using mechanistically related drugs. So this strategy should not be confused with a common combination therapy approach, where the mechanism of the disease is not fully taken into account. So network pharmacology then suggests that by targeting different proteins within the same disease model, you should expect not just a little additive effect that we see here, but a full synergistic effect. So if we are looking for synergy, then we can reduce doses and use sub-threshold doses. And related to that, we can maximally reduce potential side effects. So network pharmacology will give you the option to have minimum side effects with a full synergistic pattern. And that is actually what we are doing now. That is the direction where we are going at the moment. So using NADPH oxidase 4, so NOX4, as a seed-validated target, we found that the closest target linked to NOX4 through metabolites and protein-protein interaction was NO synthase. So NO synthase 1, 2, and 3. So then we defined a therapy where we combined NOS inhibitors and NOX inhibitors together. And we saw a reduction in infarct volume at different times after stroke, also in aged animals, and also in a permanent model. Together, of course, with some neuromotor functioning improvement, reduction of blood-brain barrier leakage, and rust formation. But the idea is that we didn't want to just focus on NOX4 and the closest targets. Actually, the idea was to extend it to the complete disease model. And for that, we defined a complete disease model from scratch where not only NOX4 and NOS 1, 2, and 3 were apart, but we also identified soluble guanylate cyclase, so SGC, and NOTS 5 as part of the same disease model. This was done, of course, using in silico bioinformatics tools. So using this actually disease model, these targets, as a pilot experiment, we treated stroke animals with a triple combination therapy, where we use NOX inhibitors, NOS inhibitors, and SGC activators. Also, as you can see here, we also had an improvement in neuromotor functioning. So here I always get a really first common question that is, okay, so what's the advantage? You had a double therapy. What is the idea of having a triple therapy? What do you gain with that? Well, actually, I can say that we saw that the triple therapy is indeed 20–30% more effective than the double therapy. Sorry. So actually, the triple therapy is indeed better, and that's where we see the difference. However, stroke is a really complex pathology, causing with several comorbidities. And one of the most relevant ones is diabetes, which normally increases the mortality rate and also worsens the prognosis. So that actually led us to generate a comorbid model of stroke and diabetes, where we were able to reduce the worsened outcome of these animals, and not only focusing on infarct size, but also we checked blood-brain barrier leakage and, even more important, survival. So, okay, let me summarize at this point of the talk, right? So at this point, we have different targets already validated, 3, 4, 5, depending if we consider the isoforms. We have a double therapy validated and published. It's out there. We have a triple therapy also validated with a potential effect in stroke and diabetes. So now what? So now what's the idea now? What shall we do now? Okay, so what we all have in mind as researchers is that, of course, we have basic science, right? We do basic science to then do preclinical experimentation, as I presented in the beginning of my talk until now, and then we move to the clinic, right? That's actually what we all have in mind as the normal pipeline. But this is indeed not what is happening in real life. In real life, we have this: a complete disjoint between preclinical experimentation and the clinics. So almost 95% of all clinical trials are conducted by industry. Almost nothing in the academia. And then basic sciences, like myself, we keep producing data, doing basic science, preclinical experimentation, publishing, and getting funding, going there again and again. And that's something that we really need to start thinking why we have this huge gap here. And on top of that, I want to actually flag another topic, and it's about the irreproducibility crisis. So actually, if we ask different pharma companies, so for example, if we ask Amgen, they think that around 89% of everything that we publish, everything that we have in PubMed, is not reproducible. And Bayer, for example, thinks that around 78% more or less. So these companies, at the end of the day, they just trust what they do in their labs, and that's it. They don't really trust that much in academia. Actually, we really need to think about that and change that as much as we can. But now, finally, I want to also make a point in our our publication system. So here we have this paper that came out a couple of years ago, where the authors divided the journals between good, here in blue, black, more or less middle, and red, not so good, based on reproducibility, based on power analysis, based on statistical power, and so on. So actually, if we have a look here, we indeed have journals where we all want to publish, including myself. We have Nature, we have Science, we have Cell. Okay, and I know this is actually a little bit shocking and tough, right? But we also need to keep in mind our way of publishing and how we are doing that. Finally, I want to move to the innovation, okay? Not only talking about publication, but also in terms of what we are able to translate. So here we have a representation of drugs that have been marketed during the last 30 years, where we have a focus here in orange in the last 10 and 20 years. So again, if we try to zoom in a little bit here, where we have the majority of the compounds or the drugs, we see that we actually moved quite fine in immunosuppressants, antineoplastic, and antiviral drugs. But there is not much more apart from this. This is much more out there. We were not able to translate drugs for other pathologies that are actually in a daily basis in the clinics. But so that's the moment that we really need to think, okay, why is this happening? Why do we have this disjoint between academia and clinical trials? Why we have this translational problem and so on? So perhaps it's related to our concept of medicine. Indeed, we have been using the same concept of medicine since the 19th century, the same disease classification or definition. So here we actually tend to systemize diseases according to body organs. So we have a specialist for every organ, right? We have cardiologists, we have a neurologist focusing on the brain, we have nephrologists, as we could divide the human body into little sections and just deal with one specific organ and understand everything about a disease, just focusing on one single organ. Or we have another option. We name a disease after a doctor, where we have, here we have, for example, Dr. Alzheimer or Dr. Parkinson, based on a specific clinical observation that they link to their names. And even more common or interesting, we actually mentioned that before, we name a disease after a symptom. So, for example, hypertension, where we know that we have an increase in blood pressure, but we don't fully understand why that is happening. So at the end of the day, we tend to treat symptoms, but not really the cause and the mechanism underlying the disease. And that's why it's definitely time to start thinking and approaching medicine in a more systematic way. And that's why I'm presenting here the network of all human diseases, the dysosome. So what is the dysosome? Okay, so here we have, in each node, we have different disease indications. And the edges connecting them are the genes that they shared between diseases. So if you actually put all human diseases and all risk genes together, we end up with this network. In some cases, like for example here, in light blue, we have a quite homochromatic cluster, including different types of cancers. Or here also in red, where we have different neurological diseases. However, if we have a look at the rest of the dysosome, it's actually quite heterochromatic. Okay, it's quite multicolor, which means that diseases that you could put in different categories, they mechanistically belong together. They share genes, they share proteins. Therefore, we are suggesting here to move far from this organ-based approach and focus on the underlying mechanism causing the disease. But reaching this point, how can we do that? Because it's easy to say, not that easy to do. So first, we actually need to integrate data like Google does with our GPS. Okay, so for example, in our GPS, we have information about postal codes, about land, about radar, about transportation. We need to do the same, but with our body. How can we do that? Integrating information about epigenomics, about genome, about comorbidities, about drug efficiency, and so on. So that's actually the next step to be able to have the complete picture of what is going on there. Here, and now I want to show you how we can actually see the dysosome from inside. So here we have disease indications, and you can also see the different edges with the different genes connecting them. So we can actually dive into it. And the next or final thing that I also wanted to show you is the interactome. So here we have information about genes, of course, and the genome, but we also have protein-protein interaction and how the different mechanisms form specific clusters within the interrectum. But okay, so what can we do with all of this? So based on the dysosome that I just presented, we indeed identified a disease cluster here, quite heterochromatic, sharing a common underlying mechanism. Diseases that in principle had not too much in common, like, for example, different type of dementias, heart failure, atherosclerosis, and so on. So sharing this pathomechanism can actually mean that they can also share similar drugs. So we were already able to repurpose a drug frequently used in cardiovascular indications like SGC activators as a treatment for stroke. But we haven't stopped there, since we are actually currently doing a similar drug repurposing strategy, but focusing on heart failure and myocardial infarction. We are currently thinking and designing 2 different clinical trials. And in parallel, we are also developing a biomarker identification strategy focused on the detection of patients suffering from this specific mechanism. So if we are able, we don't think that all patients will be actually suffering of the same mechanism, but if we are able to select those patients, we will be able to provide personalized medicine. We will be able to provide a suitable drug for them. So the idea here is to actually do it the other way around. So instead of going from basic to preclinical to clinics, the idea is to start with big data, with bioinformatics, with omics, with protein-protein interaction, and use that information and that data to be able to understand what is happening, and then go to the clinics, collect blood samples from patients, and do maybe the process a little bit from the top to the bottom in this case. So indeed, we are actually trying to do that in our stroke story that I presented in the beginning. So I already mentioned all the preclinical data that we have. Then we were able to actually do some large animal experimentation using sheep in this case. And finally, we are currently actually running a phase 1 trial, and in the next months a phase 2 clinical trial in stroke patients. So the idea is indeed to reconnect preclinical experimentation with the clinics. So for that, we need, of course, system biology, we need bioinformatics, we need multi-center animal studies, so we still need animal experimentation. And of course, a potent biomarker identification panel that would actually help us to identify proper patients. But the final thing that I want to mention is that we are not the only ones. So we are not the only ones thinking that the concept of medicine is indeed changing. This is actually from Google. So they think that they already mapped the world. They are now thinking of mapping human health. Not thinking, actually, they are doing that. And as a really fast summary, I just want to show you that, for example, we have 2 derived companies from Google, like Alphabet or Verily, where they are actually tackling age-related diseases from the in silico and big data approach. We have Amazon that actually is behind PillPack, that they want to actually take over the pharmacy market and try to actually do a sort of a modern pharmacy world. Then we have Amazon, of course, Apple. Apple is, of course, designing, moving forward with different apps where they are tracking our health data, and of course, collecting that data to be able to identify patterns, to be able to identify new patient records and use it for diagnosis and biomarker identification. And finally, I want to mention Novartis, that they consider themselves a pharma company, but a big data company, because they think that drug companies that are just treating but not healing the disease will indeed disappear during the next years. So finally, I want to thank all our collaborators. I want to thank, of course, my mentor, Professor Howard Smith from Master's University. And I want to thank you all for your attention and for giving me the opportunity to be here today. Thank you very much, and open for questions.
Anna, thank you so much. That was fantastic. Very interesting talk. So any questions from our audience?
Okay, I don't know.
Giovanni, I think you're...
I'm alive, and can you hear me?
Yes.
Yeah.
Good, good. I enjoyed your seminar very much, and I was aware of your data with NOx5. Maybe you could help me understand it a bit better. You showed that when you transfected, or I think you overexpressed NOx5 in the endothelium of the mouse.
Actually, the expression levels are really basal and not overexpression, because the animal is indeed not producing the enzyme. So NOx5 is not expressed in rodents.
It's not expressed. Okay.
No, it's expressed from rabbits.
All right. And so in that particular mouse, the stroke that you did, the middle cerebral artery occlusion and reperfusion, had limited effects in the initial radical surge on reperfusion. That's where you think the damage is, not in the neurons.
Initially, I mean, we think that it's actually happening in both sides. So we don't think it's, I mean, in a stroke, you can never say that it's something purely endothelial or purely neuronal. I think it's a combination. But I must say that in the beginning, so in really early times, the endothelium might play a key role. A little bit later, what I mean a little bit is maybe hours, okay, not days, but a little bit later, we have apoptosis, we have exocytosis, we have crazy things going on in the neurons. So, yeah, that's the idea.
Thank you very much.
Any other questions? I am very mindful of time. And I think if there are no further questions, Anna, thank you ever so much. Super interesting talk. And I'm really glad that you finally managed to present your work to us at this forum. Yeah, we're most grateful.
Thank you for organizing, and thank you for inviting me.
You're very, very welcome. So that leaves us to now engage in a public vote. Sarah, there you are. Hi.
Here I am. Hi, everyone.
Hi. So the first thing you will do is post one of the links. So can everyone go to their chat, please? And then Sarah, if you could send the first link.
It's got both on it, Alex.
So right. Okay.
Both
Brilliant. Okay.
Oral and flash talk at the same time.
Excellent, thank you. So if you could all cast your votes now, I'll be truly grateful. I can do a countdown. So we're 10 seconds from now. 9, 8, 7, 6, 5, 4, 3, 2, 1. Okay, so Sarah, do you want to announce the first and runner-up prize for the flash talks, please?
Yeah, I'm just gonna just do a final update to make sure I've got everyone's in there. So we have the winner for the best oral presentation. I'm pleased to announce is Ella Reid.
Well done.
And the runner-up is Matthew Ryan.
Excellent.
Congratulations. And then the winner for the best flash talk is Hannah Green.
Excellent.
And then we actually have a joint runner-up. We didn't
Okay.
discuss this. So that's Brenton Barrett and Chanel De Silva.
Excellent.
So
Well done,
congratulations,
everyone.
everybody.
Thank you all for voting. You can enable your microphones to applause if you want. Well, thank you all very, very much for staying on. It's really appreciated. I think from what you can all see, based on the breadth and the depth of research in cardiovascular sciences and medicine here, there's a lot that's being done. Thank you all very much for joining. It's been a really good day. So yeah, see you next year. Thanks.